Good afternoon, everyone. Thank you for joining us. This is a combined Transcend seminar with the undergraduate colloquium seminar. So we're glad you made it through the semester the year through with us. I'm so excited to introduce my friend and colleague, Lisa Tussing-Humphreys, who is a registered dietitian and assistant professor of medicine at the University of Illinois, Chicago. She earned her doctoral degree in human nutrition from the University of Illinois in 2009. Writing her dissertation on obesity and hepcidin-mediated iron deficiency. She holds a master's in nutritional sciences from the University of Illinois Urbana-Champaign and a BS in clinical dietetics from SUNY College of Buffalo. She's currently leading two-pronged research program focused on maternal nutrition, psychosocial stress, and perinatal health outcomes in diet, gut microbiome, and colorectal health cancer disparities funded by the NIH. So I'm really excited to hear her talk today. Welcome, Dr. Tussing-Humphreys.
Go ahead, Well, thank you for having me today. It was nice to get out of Chicago since we had 5 inches of snow on Sunday. So it was really welcome to have nice weather, and I got to sit outside for a few minutes. So thank you. Thanks to Meg and Corey for inviting me. And today I'm going to talk to you a little bit about our iron and pregnancy study that we conducted at the University of Illinois at Chicago between 2015 and 2017. And I'm going to start the talk just by giving you a brief overview about iron needs and regulation in pregnancy. And then I'll talk to you about some of the evidence linking obesity to iron deficiency. And then I'll move on to some of the results from our study. So this is probably a review for most of you, but during pregnancy, a woman's iron needs increase substantially. And this is really to support fetal and placental development and expansion of maternal red blood cells. So as you can see here from this slide, a woman needs about 1 gram of additional iron during pregnancy to maintain her iron balance, as well as to support the growth of the placenta and fetus. This figure here details how body iron requirements change in each trimester of pregnancy, and you can see this spans through the postpartum period as well. So from this slide, you can gauge that the iron demand isn't uniform across the gestational period. And actually in the first trimester, body iron requirements actually decrease due to cessation of menstruation. However, you can see in the second and third trimesters, body iron needs increase exponentially. And this is really in response to the expansion of the maternal red blood cell mass and rapid growth of the fetus and placenta. In order to meet the increased iron demand, both maternal dietary iron absorption and mobilization from stores need to increase and to be enhanced. And women are also advised to increase their dietary iron intake from 18 to 27 milligrams per day during pregnancy to really help meet the increased iron demand required during this period. So according to national data, so this is data from NHANES, many women actually enter pregnancy with insufficient iron stores, and they're insufficient to really meet this iron demand of pregnancy. And by the time they reach the 3rd trimester, as you can see here from this table, about 28%, what am I pressed to make this? This thing. It's light. Oh. I'm not going to use it. You can see. About 28% of women are iron deficient based on the marker transferrin receptor. They also looked at ferritin, but I wanted to point out transferrin receptor because it's less influenced by inflammation. So about 28% of women in the 3rd trimester were iron deficient, and you can see that about 11% have anemia based on hemoglobin concentrations. If you look at the bottom of the table, this is looking at across the whole pregnancy. You can see that these rates vary substantially based on racial ethnic group, with non-Hispanic Black women experiencing the highest rates of iron deficiency and anemia during pregnancy. You can see here that across the entire pregnancy, about 31% of non-Hispanic Black women will experience iron deficiency, compared to about 12% in non-Hispanic white women. And about 18% across pregnancy will experience anemia, compared to just 2.2% in non-Hispanic white women. So there's definitely racial ethnic disparities in iron deficiency in pregnancy. So the statistics I just presented are concerning, given that maternal iron deficiency and iron deficiency anemia are associated with many adverse health outcomes. Iron deficiency in pregnancy has been associated with both maternal and infant mortality, spontaneous preterm birth, low infant birth weight, some studies suggesting that low maternal iron status can affect maternal and infant immune function. And a large bulk of the literature is focused on the impact of maternal iron status on infant neurocognitive development, specifically its impact on transient and more long-term neurocognitive deficits. Also, there's been studies showing that low maternal iron status is a risk factor for perinatal depression, as well as postpartum hemorrhage in mom. And some animal-based studies have also shown that low maternal iron status in pregnancy can be a risk factor for adult onset diseases in the offspring, including hypertension and obesity. However, this hasn't been observed in humans, but this is what some of the experimental evidence has shown. So to help fulfill this increased iron demand, a woman's body adapts, allowing for increased dietary iron absorption and mobilization of iron from stores. And this adaptation is largely attributed to suppression of the iron regulatory hormone hepcidin. And I'm going to walk you through this cartoon. So hepcidin is a peptide hormone primarily produced and secreted by the liver. And it's considered the master regulator of systemic iron homeostasis. And this is because it regulates the ferroportin 1 iron exporter, which you see represented here by faucet. There is evidence that other peripheral tissues, such as adipose tissue, the heart, and the placenta, can also produce hepcidin, but it's really unknown at this point how the hepcidin produced in these tissues contributes to circulating levels. There is some suggestion that this peripheral hepcidin can have tissue-specific effects, but this is really an active area of investigation. You see here from the cartoon, again, I mentioned that hepcidin regulates ferroportin iron, the ferroportin iron exporter, which is located on tissues that handle iron in our body, including the intestinal enterocytes, as well as liver hepatocytes, reticuloendothelial macrophages that recycle red blood cells. And you can see here the placenta also has ferroportin expressed. So hepcidin itself, I want to talk a little bit about its regulation. It's simultaneously regulated by 3 signals, including body iron status, inflammation, as well as erythropoiesis. So the signals are kind of working in concert, but there are certain circumstances where one of these signals can dominate. So for example, when somebody has inflammation, but they also have concurrent iron deficiency, hepcidin may be overexpressed in this individual because of inflammation, because inflammation increases hepcidin expression. On the other hand, iron deficiency or increased erythropoiesis will suppress hepcidin. But anyway, in these individuals that have this concurrent inflammation and iron deficiency, you may have overexpressed hepcidin, which doesn't allow for the person to replete their iron stores, because what it means is that these ferroportin faucets would be turned down. So you have less iron available or moving into the circulation. So for these types of individuals, giving somebody a supplement will not really correct their iron depletion because of this downregulation of the ferroportin exporters. And we'll return to that concept a little later. So you can see here from this slide that and these are healthy pregnancies here on your left, that hepcidin changes across the gestational period actually mirror changes in maternal iron needs. Hepcidin is low or even undetectable in the late 2nd and 3rd trimester in these relatively healthy women. So this means in a healthy pregnancy, the ferroportin faucets that I just showed you are turned on high, which allows for maximum iron availability for mom and baby. So this again is when in the second and third trimester, this is for the placental formation, fetal growth, and expansion of the maternal red blood cell mass. The third trimester is also when a lot of iron is transferring to the baby for the baby's iron endowment. So again, hepcidin is very low to allow for those processes to take place. So before I move on, I just want to give you a little bit of background. A few studies have examined the impact of hepcidin on dietary iron absorption, and there's one study that's also looked at the impact of hepcidin on placental iron transfer. And these studies were accomplished through the use of stable iron isotopes. Stable mineral isotopes do not emit radiation, so they're suitable to study mineral metabolism in children and pregnant women. They are present at very low levels in our body, food, and environment, so when they're ingested, they're easily detectable in blood and body tissues. The most commonly used stable iron isotopes are 57 and 58, given their low natural abundance. And oftentimes in research, you'll see more use of the 57 isotope because it is a little less expensive than the 58 isotope. This slide shows results from a stable iron isotope study in non-pregnant women that was conducted by Kim O'Brien's group at Cornell University, which Corey was once a member of that group. The figure on the top shows, so this was a study where they supplemented women, they provided women with an iron supplement that was tagged with the iron isotope 58. And also they provided these same women with a food source that contained iron that was labeled with the 57 Fe. And you can see on the top here that it shows that as a person's circulating hepcidin concentrations increase, their ability to absorb iron from the supplemental and the food source significantly decreases. So what this data indicates is that dietary iron absorption is really impacted and is dependent on hepcidin levels. From the same group, and again, they're also using stable iron isotopes. What they showed here, and again, they provided a woman with a heme labeled iron source and a non-heme labeled iron source in the third trimester of pregnancy. And what they showed is that women at delivery that had detectable levels of hepcidin versus the women that did not have detectable levels were transferring less iron across the placenta to their babies. So again, showing that hepcidin can have an impact on the amount of iron that's being transferred across to the baby. So the take home from these couple studies, I just want you to keep in the back of your mind, is conditions in which hepcidin is overexpressed can have a negative effect on iron availability. So I'm going to take a slight turn for a minute just to discuss the connection between obesity and systemic iron regulation. So the slide that you're looking at here is from Michael Zimmerman's group at ETH Zurich. This was a supplement study where they were providing an enriched, or actually a rice that was fortified with iron, and they had also labeled it with an isotope. And what they showed here is that women, as their BMI increased, they were absorbing less of this labeled iron. So they were one of the first studies to report that there was association between obesity and iron absorption. This slide also from the Zimmerman group, not an isotope study, but they were one of the first to also show that as BMI increases, and this is in children, that hepcidin also is it's positively correlated with hepcidin. The obese children in this study also had higher levels of iron deficiency based on transferrin receptor, and they also reported that these children were more inflamed based on elevated CRP. The authors, or the Zimmerman group, suggested from this study that the adipose-induced inflammation was likely promoting increased hepcidin in these children, and in turn causing them to have a frank iron deficiency. Now again, this is a cross-sectional study, so you can't really make those causal connections, But what they did was coin this type of iron deficiency that they saw in these obese children, more of a mixed iron deficiency. So they felt that the iron deficiency that they were seeing had hallmarks of both a true iron deficiency, but also the anemia of inflammation. So with a true iron deficiency, you would see this in elevation in the transferrin receptor like you see in these children. But like with an anemia of inflammation, you also see an increase in inflammation as well as hepcidin. What's interesting about this phenotype is that we see in several studies, and one from our team as well, is that you don't see extremely elevated hepcidin like you would in the anemia of inflammation, and you do see true iron depletion. So what this suggests is that the body's able to still tap into its stores, but it's not able to replete through diet. And I think the previous study I just showed you from the Zimmerman group really shows that as well, because those women were unable, the more obese the women were, the less likely they were to replete their stores with the supplementation. At the time, so this is like 2009, 2010, many of the studies that were published were examining links between obesity, hepcidin, and iron status were largely cross-sectional. So our group sought to examine the effect of weight reduction on iron status, inflammation, and hepcidin in women undergoing a restrictive bariatric procedure. In this study, so we recruited 20 women that were planning to undergo either lap band or a gastric sleeve procedure, and assess them about a month prior to their surgery. So this is before they would have started their liquid type fast that they often do prior to these procedures. We assess their iron status, inflammation, hepcidin, body composition, a whole host of different measures. And then we invited these women back at their 6 months post-op and assess many of these same measures. And what we saw in these women is that 6 months post-op, after they lost a mean of about 50 pounds, we saw significant improvements in their hemoglobin concentrations, as well as significant reductions in transferrin receptor, inflammation, and hepcidin. So our study was one of the first to provide evidence that reducing body weight really had a positive effect on iron homeostasis. What I'd like to mention too is that our study, as well as the Zimmerman group and several other studies, were able to also show within the context of their research is that there was no difference in the dietary iron intake in the obese individuals versus the non-obese individuals. And within the context of our study, their iron intake from food and supplements per 1,000 calories was actually similar pre and post-op. So it wasn't an effect of differences in dietary iron intake. This is another study from the Zimmerman group. This is more recent. So they replicated our study and then also extended it. So they confirmed our findings, but also showed that following the restricted bariatric procedure, that obese individuals that were found to be iron deficient at 2 months post-op had increased iron absorption. So they provided these individuals with a stable isotope, and their iron absorption, it was enhanced at 6 months post-op. However, they didn't see the same trend in the obese individuals that were iron sufficient at baseline. So together from these studies, along with several others that I don't have time to review, there's many, the study suggests that in persons with obesity, that inflammation is really the dominating signal that's regulating hepcidin. So meaning that even though these individuals are presenting with true iron deficiency and it looks like they're iron depleted based on transferrin receptor and ferritin concentrations, we still see that hepcidin is overexpressed, and this is limiting their iron availability. What's important is that, again, providing iron to these individuals won't correct the iron deficiency due to the overexpressed hepcidin. However, we did show from our study and as well from the other Zimmerman study where they looked at the bariatric patients that weight loss can improve iron homeostasis in these people. So the inflammatory-driven changes in iron homeostasis we saw in the obese individuals got me, as well as many others, thinking about how obesity might affect iron availability in pregnancy, and knowing how important iron availability was during that time period, and how even subtle changes can have a negative health impact. This is a really pretty important area to research, given approximately 30% of women enter pregnancy obese, and this is based on national data, and almost 60% of non-Hispanic Black women enter pregnancy obese. So I'm just going to review with you the state of this literature, and I'm not going to go through every study, but to date there have been 10 published studies that have examined associations between maternal obesity and maternal and neonatal iron status. Findings from these studies are actually less definitive than what we see in the non-pregnant literature. So there's not a clear association in these studies between maternal obesity and maternal and infant changes in iron homeostasis. This is one of the first studies that was published. This is a case control study from Tufts University. In this study, if you look at the top first, what they saw was women with maternal pre-pregnancy obesity in the third trimester had significantly elevated hepcidin, as well as significantly elevated CRP levels. But there was really no other differences in these two groups of women. And when you look at the lower level here, this is the neonatal markers, and they use cord blood as a marker for neonatal iron status. You see here from this that serum iron was significantly lower in the cord blood of the obese women, and also transferrin saturation was significantly lower. So what these authors concluded was that iron transfer to the fetus may be restricted in the obese pregnancies. Now within this study they didn't conduct an isotope study, but this was what they inferred based on the data that's presented here. This is one of the largest studies looking at these associations, and this is a European observational cohort study that looked at the impact of maternal pre-pregnancy BMI on iron-related markers in the mom, also in the baby, and also looked at some placenta-related markers. And what they found in this study, and again, it's not as strong as what we see in the non-pregnant individuals, was that the obese women had higher hepcidin and transferrin receptor across the gestational period, and they had significantly higher transferrin receptor at 35 weeks gestation and significantly higher hepcidin at delivery compared to the normal BMI women. However, in this study, the anemia rates based on hemoglobin were similar for women across the BMI categories, and we saw there was no difference on BMI in terms of the cord iron indices. What's also interesting from these sets of studies, and I'll go over a couple more, is that there isn't really a connection to the clinical impact of these changes, and I think that's something that we're still trying to unravel, and if these subtle changes to iron status and regulation in these obese women means something in terms of clinical health impact for mom and baby. This is another study also from the O'Brien group. So they examined the effect of maternal pre-pregnancy BMI and maternal and neonatal iron status, and this was an adolescent cohort from the Western New York area. And what they found was that maternal pre-pregnancy obesity was associated with lower serum iron and higher CRP and leptin at mid-gestation and delivery compared to the normal weight moms, but they didn't see any other observable effects on the moms' iron-related indices, and they saw no effect on the neonatal indices based on cord blood iron measures. However, if you look at the bottom of the slide here, they did show that pre-pregnancy BMI at mid-gestation and delivery was significantly positively correlated with circulating hepcidin. And if you have the chance to read through this paper, kind of hidden in the discussion, they noted that when they looked and stratified on type of obesity in their moms, looking more at the severe obesity or BMI greater than 35, they showed that these women had hepcidin that was 50% higher than the other BMI groups. And they suggested that future studies should really focus on iron availability in this increasing subgroup of reproductive age women. And lastly, this is the most recent of the cohort studies examining associations between pre-pregnancy BMI and iron-related biomarkers, and this is in Mexican women, and they looked across the 3 trimesters. And what they found was that hepcidin was significantly higher and serum iron lower at all time points in women with pre-pregnancy obesity. And also, if you look at the bottom 2 graphs, you can see that transferrin receptor trended higher, suggesting that these women were more iron depleted. This study is probably the most consistent with what we see in non-pregnant populations, where you have more of that mixed phenotype with elements of both true iron deficiency and anemia of inflammation. But again, the extension then to the health-related outcomes is not available in the existing literature. So although some of the studies suggest maternal and fetal iron availability may be impaired, we don't know at this point from the literature if this is actually the case. So no iron isotope studies have been conducted to examine these questions. And that brings me to the study that I'll talk to you about today and share with you our results. So our aim was to determine the impact of maternal pre-pregnancy BMI on iron absorption of a labeled ferrous sulfate supplement in the 3rd trimester of pregnancy. And we also wanted to determine the impact of pre-pregnancy BMI on placental iron transfer of that maternally ingested iron. And we also explored how maternal obesity, pre-pregnancy obesity impacted the expression of placental proteins associated with placental iron transfer, specifically transferrin receptor 1, which is bringing the iron into the placenta from maternal circulation, and ferroportin 1, which is exporting the iron out of the placenta to baby's circulation. So in 2015, with funding from the Robert Wood Johnson Foundation, we launched the Iron in Pregnancy study at UIC. We recruited women from the UIC Center for Women's Health, their OB clinics. We were targeting women that were less than or equal to 33 weeks gestation. We wanted them to be relatively healthy, and they had to have conceived naturally and also be carrying a singleton pregnancy. We had a whole host of exclusion criteria. A few are mentioned here. We didn't want women that were underweight pre-pregnancy, that had gestational diabetes or preeclampsia, had a history of spontaneous preterm birth, because we wanted to follow these women out to their delivery. And also they couldn't have had a birth or other pregnancy in the previous 12 months. We focused on the 3rd trimester, as I mentioned earlier, because this is when the bulk of iron is being transferred to the fetus. We had 3 study-related visits. So our first baseline visit was at 32 to 34 weeks gestation. Women returned 2 weeks later to have a blood draw, and we collected some other measures that I'll mention in a moment at 34 to 36 weeks gestation. And then we also met women at their delivery to collect cord blood and placenta, as well as a maternal blood sample. We screened, no joke, about, 2,000 medical records to approach hundreds of women in the clinics to end up with a final sample of about 50 women that we were able to follow through their delivery. So it was quite a task for several years, and we had many, many people, which you'll see later, helping us with this effort. So at the 32 to 34 week research visit, we obtained a blood sample. We also dosed the women with the labeled ferrous sulfate solution. So it was 8.2 milligrams of ferrous sulfate that was labeled with the FE57. Women provided self-reported pre-pregnancy weight. We tried to confirm some of the pre-pregnancy weights if they were patients at UIC, but some of the women were new patients and were just there for OB care. So that's a caveat within our research. We also measured their weight and height, and we had women complete a variety of surveys, including a health and reproductive history questionnaire, as well as a 24-hour dietary recall. At the 34 to 36 week visit, women returned to our clinical research center, and we obtained a blood sample, and this is a sample that we used to look at their ability to utilize the iron that we provided them 2 weeks earlier. And we also checked their weight, and we talked with them about starting any new medications or if they had any new health conditions, and we confirmed these changes with the health record. And this is one of our participants in the study. She was fantastic. And my partner in crime, Mary Dawn Koenig, who is the co-PI on the study. We then met women at their baby's delivery. So just to give you some process type things that were going on in the background, the time between when we saw them at the end of the 3rd trimester visit, so that was 34 to 36 weeks gestation, we tracked them very closely. So we were in close contact by phone and text message. We also were able to work with our medical record system so that we would get alerts if a woman was admitted to the OBER, or if the woman was admitted for delivery. So we would all get like alerts on our phones. We also would look at the floors to see if anybody had been admitted from our study, and we had a 24/7 call schedule for data collection. So we always made sure that we had 2 to 3 people available each day over this 3-year period. So that was a feat in itself. Babies always come on holidays and at 3 in the morning. So that was interesting. And I think, yeah, that's all I wanted to say about that. But so when women were admitted, so usually we would get the alert from the EMR. We would meet them at the labor and delivery floor, and we would get a blood sample. So this was right when they were being admitted. And some of our women labored for about 50 hours. So this was a long process, just like waiting and waiting and waiting to get, we felt bad for the woman, but we were also very tired and exhausted waiting for the cord blood and placenta. So we, constantly in contact with the nurse midwives or with the practitioners to find out what stage of labor the mom was in. So following the baby's delivery, we obtained the clamped cord, and we harvested the cord blood right in the patient's room. And then moved the placenta to our lab space for processing. And you can see here, I took a picture of Mary Dawn processing one of our placentas. So to give you an overview of the measures, in the maternal blood at baseline and at delivery, we measured mom's iron status. We looked at a variety of different iron-related markers. We also assessed hepcidin and erythropoietin and inflammatory markers, including IL-6 and CRP. At the 34 to 36 week visit, we measured incorporation of the isotope into red blood cells. This was the marker of utilization or absorption for the mom. And then in the cord, we replicated the same measures that we assessed in the mom. In the placenta, we looked at, again, transferrin receptor and ferroportin-1 protein expression via Western blot. And we also looked at hepcidin gene expression in the placenta via qPCR. So now I'm going to share with you some of the characteristics of our sample and some of the results from the study. So as I mentioned, we recruited 50 women. 21 were obese pre-pregnancy and 29 were non-obese. The obese women in our sample were more likely to be single and not living with a significant other. And they also reported less education compared to our non-obese women. The mean pre-pregnancy BMI in our obese group was 38, which is vastly higher than what we see in the other studies that I presented earlier. So our women had more cases of severe obesity. We also saw that the non-obese women were gaining significantly more, gaining more weight in pregnancy at a higher rate than the obese women were, and this has been seen in the literature previously. The obese women also skewed younger and were more likely to self-report as non-Hispanic Black. We didn't see differences in utilization of food assistance programs, and also we didn't see differences in iron intake per 1,000 calories between the two groups. So I'm going to first present some of the maternal data that we assessed at baseline, and then I'll move on to the neonatal and placental markers. So you can see here from this slide that we saw no statistically significant differences in hemoglobin or anemia between the obese and non-obese women, although anemia skewed somewhat higher in the obese women, with just over 60% of the obese women presenting with anemia based on hemoglobin. We saw that ferritin was significantly higher in the obese women after controlling for inflammation, and also transferrin receptor was actually lower in the obese women, but there was no statistical difference between the two groups. We calculated body iron from ferritin and transferrin receptor, and we saw that the body iron was significantly higher in the obese compared to the non-obese women. So this was kind of surprising, because if you think back to the previous studies, they were in the non-pregnant populations were consistently showing that the obese women were more truly iron deficient. We saw no differences in hepcidin in the women, although it was slightly higher in the obese ladies. Not surprising, CRP was significantly higher in the obese women, though IL-6 was similar between the groups. So a little different than the existing literature that focused on looking at associations between pre-pregnancy BMI and iron and inflammatory markers and hepcidin. So our women were more obese, despite them presenting with more iron in their stores, it appeared, based on our calculation of body iron and their transferrin receptor concentrations. The obese women still had slightly higher rates of anemia. So to me and to our group, it suggests to us that these women actually had subtle anemia of inflammation. So they were sequestering iron in their stores that was likely not then being utilized for red blood cell production, given their higher rates of anemia. Consistent with this story is that of subtle anemia of inflammation is that the obese women were also absorbing less of the isotope, although not statistically less. I'm not going to repeat all of the findings from delivery, but we saw a similar story where the mom's iron status markers, hepcidin and inflammation, were similar to what we saw at baseline. So what I'm going to move on to now is the cord blood iron indices. We saw no differences in hemoglobin, ferritin, calculated body iron, or hepcidin by pre-pregnancy BMI group for the cord-based indices. There was also no difference in the amount of the isotope transferring across the placenta to the baby. Looking now at the placenta, we did see that the obese women had significantly lower transferrin receptor protein expression, and we saw that they had also trending lower expression of ferroportin protein. This was somewhat surprising to us because we didn't observe a difference in iron transfer across the placenta by pre-pregnancy BMI group. So it was a surprising finding. Given the findings that I mentioned from Kim O'Brien's study in adolescents, we wanted to just explore, we didn't run statistical tests, but wanted just to look at trends if there were differences in women if they had severe obesity. So what we see from this exploratory analysis is that women with more severe obesity, so class 2 plus obesity, had trended higher in terms of their body iron, and also the cord body iron calculation was trending lower in the women with severe obesity. When we looked at this group parsed out, we didn't really see any differences in hepcidin in the women that were more severely obese. But we did see a trend suggesting that these women were absorbing less iron, and also that less iron was transferring across the placenta to the fetus in these women. We also saw in this more severely obese group that the placental protein expression of the transferrin receptor and ferroportin was lower. together suggesting, given the trends in the iron status markers, the iron utilization and placental transfer, and the expression of these markers, that maybe these women are transferring less iron to their baby and actually sequestering more of the iron in their stores. I think given what Kim O'Brien's group presented and this very preliminary data from our study, that it is important to focus and hone in on these women and to follow a larger group of women with pre-pregnancy severe obesity to look at the effects on her iron status as well as baby's iron status, and really extend that out to look at some clinical outcomes in the baby. So to summarize our studies, obese women in our relatively small sample had higher body iron stores despite higher rates of anemia. We saw no difference in maternal iron utilization or absorption or placental iron transfer based on pre-pregnancy BMI group. And the obese women had lower expression of key placental iron transfer proteins, transferrin receptor and ferroportin-1. Our very exploratory analysis suggested that women with severe obesity may be transferring less iron across the placenta. And what we really need to understand is if these subtle changes in mom's iron status, baby's iron status, and differences in iron availability lead to any clinical outcomes. The European cohort that I mentioned earlier did recently publish a paper on their babies, and they had followed them out to 18 months. And what they showed in this paper was that independently, maternal obesity and maternal iron deficiency had a negative impact on neurocognitive outcomes in these infants at 18 months, and they had an impact on different neurocognitive domains. However, probably given the sample sizes that they would need to look at this, they didn't look at the dual burden of having both conditions, so having both obesity and iron deficiency. And I think that's really missing in the literature now to understand the dual burden of these conditions. So I really want to go through and thank everybody that helped with this study. So I'll highlight my students who I have their pictures here. So Nefertiti Hemphill, she's currently a doctoral student with me. She's conducting a study looking at the impact of the probiotic Lactobacillus plantarum 299V on maternal and neonatal iron status, and she's actively recruiting for that study. Lauren Welke, she graduated in December, and she's working at Genentech in the Bay Area. Basil Labomascus just finished her master's thesis. She utilized data from this project, and she had her own baby a week ago. So we're so excited for her. Everybody had babies in our study, by the way. Lacey Pesley, she had a baby. She's pregnant now. She is a lactation consultant. She's a registered dietitian, and she's a doula. And she is designing a mobile health intervention to improve breastfeeding outcomes for her dissertation project. I also mentioned that I had a partner in crime, Dr. Mary Dawn Koenig. She's a nurse midwife from our College of Nursing. She had amazing staff and midwives that worked with us as well. Dr. Kim O'Brien at Cornell did the isotope work for our study. And my mentor and colleague from UCLA, Ellen Nemeth, helped us with the placental protein analysis. And then our funding is there. And then I have to thank all of the moms and families that let us lurk around in their rooms while they were having a baby, and were just absolutely amazing. And we had just, I really think, fantastic retention for this type of study. So we were able to follow 85% of the moms through the delivery. Most of the babies, the deliveries that we missed is because moms went by ambulance to a different hospital. So the moms were really engaged in our study, which was amazing. So thank you again for having me, and I'll take any questions.
Open it up for questions.
I have a question. So you said it was restricted by inflammation. Did you guys look at other factors of inflammation in their diet that could have been improving?
That's a good question. So in terms of Are you talking like a dietary inflammatory score or something like that?
When I eat gluten, I get inflamed, my joints get inflamed,
Mm-hmm.
and you said that that restricts the iron
Mm-hmm.
going into the blood. Did that affect any of the moms in your study?
So we didn't calculate a dietary inflammatory score, which we certainly could. We did look at some of the known inhibitors and promoters of iron absorption to see if there are differences between our two groups of moms, as have other studies, and we didn't see differences between the obese and non-obese moms. So that's a good question.
Are there any hepcidin pharmacological or nutraceutical antagonists or inhibitors?
Not on the market that I'm aware of, but definitely under development. Dr. Ellen Nemeth that I mentioned at the end there, she's involved in some of those pursuits.
And so you're seeing these women later on in their pregnancy, did you look at their prenatal vitamin compliance, or would that even make a difference since you were looking at the transfer like within?
We didn't look at their compliance across the whole pregnancy. We collected some data about recent use through the 24-hour diet recall. And we actually had them refrain from their vitamins for 48 hours before we provided the isotope, because that could increase their hepcidin levels and could have affected that process. But I don't know if it was so important in the context of our study, but in the study that my student's doing right now, Nefertiti, she is tracking very closely. We're actually using smart pill bottles to look at compliance with the prenatal vitamins as well as the probiotic that we're giving.
Did you guys ever do like a gut biopsy or anything preliminary research? To see if microbiome is such? Did you do a biopsy?
No, so in the moms, we collected stool samples and we did do some 16S analysis on those moms. And we didn't look at it by obesity because we didn't see anything too interesting, but we saw when we combined the two groups of women, the obese and the non-obese, the women that were absorbing less iron, their microbiome looked different than the women that were absorbing iron, and that's something we hope to follow up on, but that's a good question. In the bariatric studies, we weren't able to obtain intestinal samples, but we did collect adipose tissue samples from those individuals, and we looked at expression of hepcidin within the adipose tissue.
So you mentioned that the isotopes are, and I don't think a lot of people are aware of how expensive isotopes are and their different prices. Could you ballpark share the difference in?
I can't remember what the total amount of isotope was that we purchased, but I think it was like $7,000. For like the difference. I think there's like a 30% difference in terms of the cost. So we used the FE57, and that was about, I don't know, anywhere between $5,000 and $8,000. I can't remember.
Per participant?
No, total. Yeah, so that was enough for our 50 participants.
Any other questions? Well, let's thank our guest again.
Thank you.